First of all, let's begin with the interphase. The interphase is where the normal processes in the cell happen, and then after this comes the prophase. During interphase, before the start of the prophase, chromosomes are replicated and consist of two identical sister chromatids joined at the centromere. In prophase 1, these chromosomes now supercoil and the homologous pairs of chromosomes line up side by side. The final step of the prophase is the breakdown of the nuclear envelope. After this, the centrioles go on opposite sides of the cell. The chromosomes line up on something called the equator. The alignment of the chromosomes is random so that the maternal and paternal chromosomes can appear on either side of one another on the equator. This is the beginning of metaphase 1. To create genetic variation, something called chiasmata are created. This is where limbs of separate chromosomes cross over and swap some genetic information. The spindle fibers from each centriole then attaches to the centromere of each chromosome. The spindle fibers now contract towards opposite poles. The pairs of sister chromatids remain together, but the homologous pairs are separated. This is the reduction division where the chromosome number is halved from diploid to haploid. This begins anaphase 1. Now spindles break down and a new nuclear envelope forms around each new nucleus. Cytokinesis follows and the cell splits into two cells, each containing only one chromosome of each homologous pair. Each chromosome, however, still consists of two sister chromatids at this point. The second division of meiosis now follows to separate the two sister chromatids. This ends telophase 1. Now begins prophase 2. In each of the two cells resulting from meiosis 1, new spindle microtubules start to form. The chromosomes recoil and the nuclear envelope begins to break down. Now begins metaphase 2. The nuclear envelope is broken down and individual chromosomes line up on the equator of each cell. Spindle fibers from opposite ends of the cell attach to each chromatid at the centromere. Now begins anaphase 2. Sister chromatids are separated as the centromere splits and the spindle fibers pull the chromatids to opposite ends of the cell. And now finally, telophase 2. Nuclear envelopes form around the four new haploid nuclei, and the chromosomes now uncoil. A second cytokinesis occurs, resulting in four new, genetically unique haploid cells. This is where you and I come from. And now you know a little bit more about meiosis. Thanks for watching.